Hello everyone. Today we are here to discuss a question generally asked as a long question in MBBS second prof. The question is define necrosis, its causes, pathophysiology, and explain in detail types of necrosis. So basically in this video I am going to tell you guys how to write this question in prof examination and secure maximum marks. So we will start with the definition. Necrosis is a pathological process that is the consequence of the severe injury. It is a form of pathological cell death. Now let's move to the causes. The main cause of necrosis include loss of oxygen supply, which means ischemia, exposure to microbial toxin, burns, and other form of chemical and physical injury. Now let's move to features. Necrosis is characterized by first, denaturation of the cellular protein, second, leakage of cellular contents through damaged membrane, third, local inflammation, and last, enzymatic digestion of the lethally injured cell. Now, after completing causes, definition, and feature, now let's move to the pathophysiology or mechanism. In general, I would recommend to write mechanism or pathophysiology in the form of flowchart and draw some necessary diagram if possible. So we can write. So after completing causes, definition, and feature, now let's move to the pathophysiology or mechanism. In pathophysiology or mechanism, it is generally advised to make flowchart and diagrams if possible, as I have made here. So if we talk about mechanism of necrosis, when damage to the membrane is severe, it would lead to the lysosomal enzyme enter the cytoplasm and digest the cell, as I have shown here. Eventually, cellular content also leak through the damaged plasma membrane into the extracellular space, where they can elicit a host reaction. As I have shown here, there are some substances which are released out of the cell. Some specific substances released from the injured cell are known as DAMPs, DAMPs, damage associated molecular patterns, which include ATP, uric acid, and troponin. ATP are released from the damaged mitochondria, and uric acid is released as a product of DNA breakdown. These DAMPs molecules are recognized by the receptor present on the macrophages, which will trigger phagocytosis of the debris as well as production of the cytokine that induces inflammation. As I have shown here, the macrophage is recognizing DAMPS molecules, which and ultimately releasing cytokines, which is causing inflammation. And if inflammation persists, it will lead to chronic inflammation, which will ultimately lead to necrosis. Now let's move to the clinical. Adding clinical correlation can provide you an extra edge over others. So here we are going to discuss clinical of the necrosis. Necrosis is associated with the leakage of intracellular protein through damaged plasma membrane and ultimately into circulation, which is the basis for tissue blood tests that detect the tissue-specific cell injury. In heart, we have cardiac muscle cells, which expresses cardiac-specific variants of the contractile protein known as troponin. In hepatobiliary system, bile duct epithelium expresses a specific isoform of enzyme known as alkaline phosphatase. Hepatocytes expresses transaminases. Necrosis of all these types of cells associated with the loss of membrane integrity is reflected in the increase of the serum level of these proteins, which serve as a marker for tissue damage. Now let's talk about microscopy. In microscopy, we can write cell necrotic cells show increased eosinophilia in H&E stains, attributable in the parts to the loss of cytoplasmic RNA and in the parts to the accumulation of the denatured protein, which bind to the red dye eosin. The necrotic cells may be glassy, homogeneous appearance relative to the normal cell, mainly as a result of loss of glycogen particles. When the enzymes have digested the cell organelle, the cytoplasm becomes vacuolated and appears as moth-eaten. Dead cells may be replaced by large whorled phospholipid precipitate known as myelin figures. They are spiral in shape. Myelin figures, further phagocytosed by the other cells, or further degraded into fatty acid, and which upon calcification of such fatty acid residue result in the calcium-rich precipitate. By electron microscopy, necrotic cells are characterized by the presence of large amorphous densities in the mitochondria. Now coming to the types of necrosis. The first type of necrosis is coagulative necrosis. It is the form of necrosis in which architecture of the dead tissue is preserved for a span of at least some days. Coagulative type of necrosis is most common type of necrosis. Injury denatures not only the structural protein but also the enzymes. So it blocks the proteolysis of the dead cells. Ischemia caused by the obstruction in a vessel may lead to coagulative necrosis of the supplied tissue in all organ except brain. A localized area of coagulative necrosis is called an infarct. Second type of necrosis is liquefactive necrosis. It is characterized by the digestion of the dead cell, resulting in transformation of the tissue into a liquid viscous mass. The necrotic material is frequently creamy yellow because of the presence of dead leukocytes, and it is called pus. For unknown reasons, hypoxic deaths of cell within the CNS often manifest as liquefactive necrosis. So if any necrosis occurred to brain or CNS, it would be generally liquefactive type of necrosis. Now moving to gangrenous necrosis. Gangrene is basically there are two types of gangrene, a dry gangrene and a wet gangrene. As the name suggests, dry gangrene, it means it will be in dry in appearance, and gangrene will be liquid in consistency or liquid in appearance. Generally, gangrenous necrosis generally occur to a limb, generally the lower limb, that has lost its blood supply and has undergone necrosis involving a multiple tissue plane known as dry gangrene. When the bacterial infection is superimposed, there is more liquefactive necrosis because of the action of the degradative enzyme in the bacteria and the activated leukocyte, giving it a wet appearance, also known as wet gangrene. Now let's talk about the caseous necrosis, generally seen in tuberculosis infection. It is generally called caseous because of its cheese-like appearance, cheesy because of the presence of mycolic acid. On microscopic examination, coagulative necrosis and liquefactive necrosis are seen. Amorphous granular pink structure are also seen on microscopic examination of caseous necrosis. Now, next type of necrosis is fat necrosis. It may be due to first enzyme, second trauma. There are basically two causes of fat necrosis: enzymatic cause and traumatic cause. Any trauma to the breast injury may cause fat necrosis. And when we talk about enzymatic, in acute pancreatitis, lipases are released, but due to this disease, lipases are activated inside the pancreas itself, which will act on lipids and release the fatty acid and combine with the calcium, which is called saponification. And on saponification, it will lead to the gross structure becoming chalky white appearance. Next type of necrosis is fibrinoid necrosis. Fibrinoid necrosis occur due to the presence of fibrin plus immune complex. Immune complex is a product of antigen and antibody reaction. So when fibrin and immune complex are together, it will lead to the fibrinoid type of necrosis. What is its pathology? For example, if a vessel wall is damaged, it will lead to the coagulation pathway, and coagulation pathway will lead to the fibrin formation. And if this fibrin comes in contact with the immune complex, it will lead to the fibrinoid necrosis. It is seen in vasculitis such as polyarthritis nodosa and malignant hypertension. If you see pink amorphous and homogeneous substances in a blood vessel, you can suspect fibrinoid type of necrosis. This marks the end of the video. In this video, I have told you how to write answers of long question on necrosis. You may use the answer exactly as it is prepared 100% from Robbins. So if you like the video, Please do like this video and share and subscribe our channel. Thank you so much for watching this.